Great. Hey guys, so now I wanted to talk you through on some more images from the board from class. These images are going to are going to show us how the sperm finally finishes its development inside the female womb and how this sperm is able to ultimately fertilize the ovulated egg. So if we switch over to this image. Okay, so here's an image that shows sperm that has already entered into the womb of the female. This sperm has been able to travel in through the uterus and it enters into the oviduct. You'll notice that it has a head, it has the mid piece, and it has a flagella, this green area, that's the acrosome. And these red little proteins, those are proteins that are on the inside of the acrosomal membrane. That'll come in later. But this sperm, even though it's entered into the oviduct, it's still immature. And it has to go through a process called capacitance in order for it to be able to fertilize an egg. Here's how this works. So this immature sperm is going to get stuck to the cells on the wall of the oviduct. This is absolutely necessary for it to complete its maturity. After this happens, that's going to initiate a process called capacitance, which will cause the sperm to get really active. And it's going to start beating its tail like crazy. And that beating of the tail is going to cause it to break away from the cells of the oviduct. And it changes this process of like binding to the cells of the oviduct. It's going to kind of change the chemical properties of the acrosome and the cell membrane of the sperm, which will make it easier to go through later steps. So we have these like distinct physical changes in the head of the sperm, but also it becomes really active. It starts beating its tail like crazy, and that will release itself from the wall of the sperm. After this happens, it's considered to be capacitated, and it is a mature sperm. Now, a capacitated sperm or mature sperm is now able to go through what's called the acrosomal reaction. This is a process where the acrosome, basically the membrane that holds the acrosome, reverses itself. So here in this sperm, you can kind of see how the green acrosome is on the inside of that acrosomal membrane, along with those red proteins. They're on the inside. This acrosomal membrane kind of folds in on itself so that those red proteins are now on the external surface of the sperm, like the inner membrane of the acrosome is now exposed. This is absolutely necessary for the sperm to be able to fertilize the egg. So now, after it's gone through the acrosomal reaction, it is now capable of fertilizing the egg. So now let's go to the next image here. So what we'll find in the next image is, okay, great. So here is a picture of an egg that has just been ovulated. Notice you have the purple oocyte. You're going to have the blue zona pellucida, which is the layer of thick glycoproteins that protects the oocyte. And then you also have the corona radiata, which are follicular cells that came with the oocyte after ovulation. So what will happen is that we have hyaluronidase, which is an enzyme in the cell membrane of the sperm's head, which dissolves hyaluronic acid, which is holding these follicular cells together. So these brown follicular cells are held together with this hyaluronic acid. We have an enzyme in the head of the sperm that will dissolve these connections between the cells, allowing the sperm easy access to the deeper layers of the follicle. Now, the next job of the sperm is to bind onto special proteins in the zona pellucida. And these proteins are ZP2 or ZP3. ZP2 is going to bind to proteins in the head of the sperm, and this is like the lock and key mechanism. If the proteins of the sperm cell membrane do not match up to the ZP2 protein of the zona pellucida, the sperm cannot enter, and this is one way in which the egg can prevent itself from being fertilized by sperm of a different species. Okay, it's only sperm with the correct proteins are going to be able to bind. Now, if sperm have not undergone the acrosomal reaction, then the acrosome will bind onto ZP3, which will in turn trigger this acrosomal reaction. That should have already happened, but if it didn't, it'll trigger it, and that would then allow the sperm to then bind to ZP2 and make its way through this zona pellucida. It not only binds to ZP2, but as it binds, it kind of makes its way deeper and deeper through this zone. Finally, the sperm gets access to the wall of the oocyte, the cell membrane of the oocyte. There's two proteins that are going to join together, which will allow the oocyte to absorb the sperm. You have these orange Juno proteins, which are on the surface of the egg's cell membrane, and these red Izumo proteins, which are on the surface of the sperm. Now, the sperm kind of lays on the oocyte sideways, and after these proteins unite, the oocyte will absorb the sperm, the head, the midpiece, even the flagella. After this happens, we will a very distinct change occurs quickly in the cell membrane of the egg. This is going to prevent other sperm from fertilizing the egg, and this is associated with the release of calcium that causes the zona pellucida to become impenetrable by other sperm. That's why we have this black zigzag line going through it. This happens after the sperm begins to be absorbed by the oocyte. Now, after this happens, the only thing left to achieve is that we have the fusion of the pronuclei of both the egg and the sperm, and this will ultimately produce the zygote. So